environment in the biology.
Ooh, sorry.
Oh, that's fine. Within the marine biology department, that our master students are defending their proposal, so their plan on the research that they are doing. And so that's what Charlotte is going to do. So she's going to tell us a little bit about, kind of the work that she has been doing on sharks. Um, and just to give you a little bit of background on Charlotte, so she has an associate's degree from Cascadia and then went to UA Southeast and got a, bachelor's in marine biology. And so, yeah, and then she joined us in the marine biology department, and that's what she's doing right now. She has done tons and tons and tons of, kind of different projects that she's been working on. So a lot of fun things, and that's pretty much where this particular project has led to, is because Charlotte was working with Noah and Cindy on this very cool project on sleeper sharks and their life history. So Charlotte, take it away. And then if I could ask everybody else to hold their questions until the end, I will monitor the chat, but you can obviously also ask Charlotte herself. All right.
Okay, thank you, Laura. I appreciate it. Um, Everybody in the room, make sure your phones are on silent. All that good stuff. Everyone on Zoom, you all look muted, so we're good to go. Okay. Well, we're just gonna jump right into it. As Laura said, this is gonna be my proposal presentation for my master's thesis in marine biology. And we're gonna be talking about stable isotope analysis of Pacific sleeper shark islands laminae, to hopefully reveal some lifetime trophic ecology. So really quick land acknowledgement. I think it's important that we acknowledge that we reside on the unceded territories of the Akwon, also known as Juneau, Alaska. Tlingit peoples have been stewards of this land in which we reside and work since time immemorial, and we're very grateful for that stewardship. It's a beautiful place, and we've had a beautiful summer here. So really quick, going through the core team here. Charlotte Springer, that's me. Hello, I'm the grad student. Laura Horseman is my advisor. Cindy Trujillo is my external committee member and prior NOAA boss and still mentor and close colleague. And Matt Wooler is my other committee member who's isotope guru and also great mentor. So throughout this presentation, we'll kind of refer back to this navigational slide as kind of a reference point of getting your bearings of where we're at throughout the presentation. You'll see that there are slide numbers throughout this presentation as well. So if you guys have questions on a specific slide or tied to something on a slide, that slide number will be there as an easy reference point to hopefully go back to. So starting out with our background and where our story begins, who we're working with. We are working with Somniosus pacificus, the Pacific sleeper shark. Here we have one that's actually been spotted down in Antarctica, who's flipped upside down. He's trying to take a bite out of some bait. His eye is, let me find my little cursor here, right here to orient yourself, and there's his mouth. So it's kind of an upside down torpedo. But throughout the background here, we're going to kind of talk about their range, which is the North Pacific subarctic region. We're also going to talk about a little bit of their habitat that we know, benthopelagic, the slopes. They're a deep ocean species. We're going to try and touch base on population dynamics. There's not much known about that. We're also going to try and touch base on trophic pathways as well as migration, age, a lot of the stuff we don't really know anything about. So it'll be very quick. And their current status is near threatened. So where does this all start? What do we know? We know that they're caught pretty prevalently in North Pacific fisheries as bycatch, and that is where we get a lot of our data from. With that in mind, their ecological role is still poorly defined. And this raises questions like, are they competing with groundfish for food and with us in fisheries? Are they being caught coincidentally? Are they happen to be existing in the same areas as those fisheries and they just happen to be, so to speak, in the way, but not in the way, you know? So looking at their known distribution, as I said, their known distribution is throughout the North Pacific. All along the coastlines, they've been spotted, usually caught as bycatch. They also bleed up into the subarctic ocean. this is more of a distribution that shows where we know they are, not where they aren't. They could be throughout the entire Pacific for all we know. We just don't have enough data. Has been spottings of them in the southern Pacific region. There's current research, genetic research being done on whether those are a subspecies or the same species. We don't know. So for the purpose of this, we're focusing on the North Pacific. Overlapping this with a little bit of bycatch data. This is from 1997 to 2021. Bycatch in metric tons of Pacific sleeper sharks, caught mostly within the Alaskan fisheries region. You can see that we have a prevalent amount of them in the Bering Slope region, as well as the Aleutian Islands and the Gulf of Alaska range. They're all throughout here. So it's a very similar distribution to what we're seeing in the distribution map, which comes from it mostly being fisheries data. So what do we know about their diet? What we know is from 2 individuals that have had visual stomach content analysis done. So take this with a grain of salt because it's only 2 individuals. They have found a variety of things within those 2 stomachs. From rockfish, ground fish species like halibut and sablefish. They've found king salmon. They have found copious amounts of beaks belonging to various cephalopods, squids, octopus, you name it. They have found evidence of other sharks, as well as various types of carrion belonging to seal, moose, whale falls, pretty much anything they can find. So we can assume that they are probably pretty active scavengers and potentially active hunters as well. Moving into the more broader strokes of what we don't know about sleeper sharks, what's unknown about them and why don't we know it? Well, traditional methods for studying their feeding ecology and movement patterns, it doesn't work very well with sleeper sharks. Reasons for that is they're deep sea, they're large, they don't really handle mark recapture well at all, as in none of them have successfully been re-released back into the ocean, so tagging them hasn't worked very well. They don't handle that, being brought up from depth and being re-released. They're usually dead on arrival sort of deal. So again, that creates gaps in our knowledge in terms of what is their interactions with fisheries species. What is the timing and frequency of their feeding habits in terms of magnitude, consistency? We just don't know. And movement patterns, are they migratory or stationary? We know they have a really broad range, but whether they're different groups hanging in different ranges or groups moving throughout the entire range, we don't know. So there's some key questions here that we're looking at that we're hoping to kind of narrow down and answer with this study/project. Are there ontogenetic shifts in the habitat that indicate shifts in their ecological role? Meaning, do they move throughout their life, basically? And if they do, how does each location during their life, how do they impact each location in that stage of their life? What trophic levels do these sharks occupy throughout their life? And with that, what is the potential influence on fisheries species? Of course, that's the big question that everyone in management wants to know. So same thing, are they changing trophic positions as they age and potentially move? We don't know. So tying this in with some relevance to some bycatch data, resolving the ecological role for these long-lived predators, it's increasingly important for the interpretation of the bycatch. Understanding their species distribution as well as that predator-prey dynamic, it's incredibly important for informing commercial fisheries management and all the stock assessments, how all that's managed. We need to understand what influences these guys potentially have on our fisheries fish that we want. So looking at this figure here, we're looking at catch rates of sleeper sharks within the Bering Sea and Aleutian Islands on top, Gulf of Alaska in the middle, and then Southeast Alaska / Prince William Sound on the bottom. You can see they're mostly caught within the peacod and flatfish fisheries. Sorry, I can't read it from here. Can't remember off the top of my head. And then Southeast Alaska, it's caught more within various individual fisheries that are undescribed here. And that is from data that is coming more recently from 2013 and on. The rest of this data is from 2003 up to 2021. So a few gaps on our front end here, but it gives a good perspective of sleeper sharks that were caught and kind of see a decline throughout the years in terms of abundance that are being caught. Taking a quick look at the gear types that they're mostly being caught by, you can pretty much ignore the pot. The big things to take away here is that they're mostly being caught by trawl and by longline. Again, we didn't have data for longline until after 2005. So it's not to say they weren't being caught by longline before, it's just we didn't have data for it. But the longline data is in numbers of individual sharks, while the trawl data is caught in metric tons. So way more caught by trawl than by longline, though there's still a significant portion that are being caught with both. So a little bit of perspective there on how much they're being caught as bycatch. Last graph, I swear. This is looking at the indices of abundance within different regions, as calculated by 4 different organizations. It's looking at data from NOAA, so National Oceanic Atmospheric Administration, the DFO, which is the Department of Fisheries and Oceans. We're looking at ADF&G, Alaska Department of Fish and Game, as well as the IPHC, which is the International Pacific Halibut Commission. The crux of this is showing pretty much a decline in sleeper sharks in all regions, with exception of the NOAA Bering Sea Shelf Trawl Survey, where they are doing okay. Very sporadic, but they aren't going down, which is interesting in itself. But the crux of this is we're seeing a decline in all of these, with exception of that one, which is why they're currently noted as near threatened. So how does understanding their lifeline habitat use in feeding patterns, like what will that help clarify for us in terms of North Pacific ecosystems and fisheries and stuff like that? Well, trophic positions if we can figure out anything with the isotopes of the sleeper sharks, it will help understand the trophic positions within North Pacific food webs. It'll help us understand how trophic roles may change across life stages. And it should help us figure out whether they are migratory or stationary. And so where does that leave us? Mr. Sharks asks. That's going to bring us into the project itself. What are we doing and what are we doing it with? So we are going to learn a little bit about isotopes and a little bit about eyes. So really quick lesson on isotopes for those who are not familiar. Organisms, all of us, anything living, experiences what we call fractionation of isotopes. That is where you have a heavier isotope, for example, 13 carbon, which has an additional neutron, compared with a lighter isotope of 12 carbon. One will stay within the system more readily than the other. So the lighter isotope, 12 carbon, will flush through a system more so than 13 carbon, and 13 carbon will be more apt to stay. That becomes a ratio that we can look at that we would call delta 13C values. It's the same scenario with 15N compared with 14N, noted as delta 15N values, and they can tell us different things. So delta 15N can inform us on relative trophic position, while delta 13C values can give us insight into habitat or carbon source shifts. Hopefully that makes sense. So how can we utilize delta 13C and delta 15N values within sharks themselves? We have to have something that we can pull them out of, and that is where it gets a little tricky. Sleeper sharks all sharks lack any calcified structures that lay down rings like otoliths, because sharks don't have otoliths. Teeth don't work very well because they slough them off, and you could use them for a timestamp, one singular timestamp in the shark's life, but it would not work for an ontogenetic study where we're trying to figure out what's happening with them throughout their entire life. Some studies have used their cartilaginous vertebrae. It's kind of given muddy results. The data is a little inconsistent and it's hard to replicate because their vertebrae are cartilaginous, so it's hard to get distinctive growth rings that are consistent. So that leaves us with eye lenses. So the eye lens itself is composed of metabolically inert protein layers, which can preserve a chronological archive of isotope values. Each of those layers is going to contain carbon and nitrogen from the diet during the time in which it was formed. So it creates a perfect timestamp for us to go back and refer to. There are things to consider when you're looking at different tissues within any organism with isotope analysis. One of the big things in most cases when you're working with muscle tissues is things like lipids. How can that influence the isotope ratio? We aren't too concerned with that with isotopes in eyes because there is really no lipid structure within the eye lens itself. So we're not concerned with it there. We do want to think about urea though, as this is a heavily uritic shark species, as most of them are, especially a deep sea sleeper shark. Um, Based on other research that's been done, we found that flushing the eye lenses with water removes any potential impact on urea from urea. So we aren't too worried about that one either. Okay, we're going to do a little bit of eye anatomy lesson for those who are not familiar with them, which I wasn't when I first started this project. So it's been fun to learn and it's pretty easy. So bear with me. On the left side here, we have a full eye with a cross section showing where the eye lens sits inside. So on the top side, you have I guess I have a little pointer here, I could use it. We've got the cornea and the iris here. Top of the eye, this is what you would see on a living shark, usually where the parasitic copepod likes to hang out. Within the eye itself, you would have this vitreous humor fluid that holds this capsule down at the bottom, and it would also be connected to an optic nerve here, which I did not illustrate. But that is where it sits. If we zoom in closer on the eye lens itself, it is composed of pretty much three main components of three biological components. We have the outermost layer, which we would call the capsule, and you can think of as a water balloon. It's like a membrane that holds everything together. We have the secondary layer called the pre-apoptotic crystalline proteins. So these are quote unquote living proteins that are currently uptaking stable isotope values at that moment in the shark's life. Those proteins will eventually go through a process called apoptosis, where they will harden and form the next hard layer on that lens core, which is this innermost part here. And then we can zoom in a little bit closer to the lens core itself, where we have all these layers of crystalline proteins that are an archive of the shark's life for us. And so that's our bread and butter. That's what we want. It's going to be really cool. And with that, now that everyone's a master of eye biology and isotopes, we can take a look at the objectives and the hypotheses for this. So we have three objectives that we're working towards. The first objective is pretty simple. It's develop and validate the eye delamination methods. So can this be done with sleeper shark eyes? It's been done with other teleost eyes, but can it be done with a deep sea creature? Pretty much. The second objective is to look at those delta 15N values. So identify ontogenetic trophic level changes by analyzing those delta 15N values across sequential eye lens laminae. So is there any distinct trend? Is there a trail a story that's being told with the data that we're getting with the delta 15N values? And the third objective is pretty much the same thing, but with delta 13C values. We want to look at and try and reconstruct the lifetime habitat use and carbon source dependence by analyzing those delta 13C values across the eye lens laminae. So see what we can get with it. Paralleling those three objectives, we have two hypotheses that pretty much say the same things. The first one being delaminating sequential eye lens laminae will provide bulk delta 15N and delta 13C values that indicate identifiable ecological shifts consistent with ontogenetic change, as occurs with other marine predators. So we should expect, our hope is that we would see a similar trend like we do with other marine predators like sailfin fish and tuna and stuff like that. The second hypothesis is that sleeper sharks will exhibit the ontogenetic and individual variation within trophic ecology, which again would be expressed as increasing delta 15N values from the innermost, so the youngest point in the shark's life, to the outermost laminae, the oldest point in the shark's life, which would be indicative of moving to higher trophic levels as they age, and increases in delta 13C values that reflect a shift from shallow to deep water ecosystems. So our current hypothesis is they might be starting shallow and moving deep. Okay. We've established the goals. Let's move on to methods, samples and methods. So this is where all of our samples are coming from. Each of these marks is an individual shark. This data is all the way from 2011 to 2025. So we have around 200 sharks that we've used for this study. We obviously don't have all of them processed, but we have that many in our sample database that we could potentially run samples for. Just as an aside, it's interesting to see that we have mostly smaller sharks in the Bering Slope region here, Bering Sea, and we have larger sharks all along the Aleutian Islands, Gulf of Alaska, and Southeast Alaska. So there's an interesting discrepancy there that, there's not enough data to make any strong conclusions on, but is interesting nonetheless. So that's where they all came from. How are we getting them? So as I mentioned, a lot of these guys are coming from bycatch off of trawlers as well as long lines. So trawling surveys, long line surveys. We have observers on those vessels that are collecting the eyes from us as well as all the other data. We're also collecting eyes and data from strandings that we can get to. So we've had a few strandings here in Southeast Alaska. The one you're looking at here is actually from Eagle Beach here in Juneau from 2024, as well as local catches. So local fishermen sometimes give NOAA a call and say, hey, we have a shark. Do you want it? And we usually say, absolutely. So yeah, 6-foot person to 10-foot shark. That's a pretty crazy comparison. I believe this one was caught back in 2011. So this is one of our first sharks that we've had in our database for a long time. Okay, once we have the sharks / the eyes and all the data, we can start the dissection process. This is where it gets gooey and smelly and fun. So apologize to all the other lab people that had to deal with the smell of the shark eyes, but it's a pretty easy process. So you want to go through the process of dissecting the eye, which entails cutting open the cornea delicately. So you don't want to cut too deep so you don't cut the lens on accident. Once you've cut it open, you can start scooping out the lens, which is sitting down at the bottom of the eye here. And once you scoop it out, you have your lens and you can start the delamination process, which is long and arduous in some cases, and it's very delicate. So this is all done under a microscope, making sure we're getting consistent peels that are not too thin or too thick, and they are consistent all the way around. Will Litch teaching me some tips and tricks down here on how to do it with the teleost eye, and then incorporating those methods into sleeper shark eyes. So in the top left here, you can see that capsule layer is really shiny and gooey. And once that peels off, you have that pre-apoptotic, which is also very gooey. And then from there, it turns into a more hardened crystalline layer. And that is that lens core that we peel down as far as we can go until we get to the smallest point without it fracturing on us. So depending on the size of the shark, we could get different size peels or number of layers peeled off of them. And this is just a closer look at the peels themselves of what we're looking at under the microscope here on this side, and an illustration of what I'm doing under the microscope. So using some tweezers, usually it's 2, and we're sitting there trying to peel them apart very delicately. With most isotope processes, once we have the sample, it's filed, it's weighed, it's dried till it's consistent, till it's unchanging. Once it's completely dry, we seal it, and in our case, we sent them off mostly to Stanford for stable isotope analysis. They were also sent off, a subsection was sent off to the ASIF lab, the stable isotope lab up in Fairbanks for class project, and also as just a subsample. So we have some data we can compare there, which is interesting. And we also did send a subset for compound specific, which we'll get into in just a bit. And so this slide is all funding dependent, which currently we don't have, but one of the goals with this project was to run a larger subset of samples at UAF with the ASIF labs, in part for training for myself on the EAI IRMS system and learning the tail end of that process. And in turn, that would also give us some more results that we, one, have more data, 2, we could compare lab to lab and see. I wouldn't expect there to be any discrepancies, but we could see. So that was a potential thing that we could do. Okay, that was crux of the proposal. And since we've done a lot of this stuff, we do have some preliminary data. So a lot of this is going to be bonus content from here on out. So hopefully it's all a work in progress, I will preface. So there's certain things you might think are a little muddy. It's because they are. and we're working on narrowing it down and, cleaning it up. So here's our current data that we have. We have a total of 66 sharks that we can use in terms of actual analysis. They had enough layers, their data came back clean, and we have enough metadata for them that we can put context behind the data that we got. We have a pretty relatively even distribution between male to female. We had a few unknowns. Whether we'll keep them or not, we aren't sure yet. Same thing with size ranges. We got a pretty wide variety of size ranges of sharks. So we could assume different sizes, means different age ranges. And then layers per shark. Larger sharks, we got more layers out of in terms of laminae peels. And smaller sharks, we didn't get as many out of. So some of those smaller sharks will probably get removed just based on how many layers that we have to work with. Okay, this is where it's exciting and we can actually see some of the data. So this is looking at the delta 13C values compared to eye lens diameter in millimeters. And this is 66 of those individuals graphed out all on one and with a smoothing line, so it can make it a little more easier to see what's happening. There's something happening here, which is really exciting. It's not muddy, they're showing a clear path here. What does it mean? We're still kind of working on figuring that out. One of the things that we've talked about, which we can't confirm right now, but is one of our current hypotheses, is if this is a potential maternal influence. So this first slope here is really interesting because we're like, why does it drop down and then start climbing back up again? One of those things, like I just said, could be that this is from the mother. And then at the bottom here is time 0 for our individual sharks. And then that is where they're born. And then they're starting their own isotopic journey per se. So that is one potential way to interpret this. And we're looking at other ways too. So looking at the delta 15N values compared to eye lens diameter, it's a little different and not as what we expected, but we do see a climb, which is really interesting. And we do see steps in it. So we're still working on, how can we understand this? Like we can the delta 13C values and all that. It shows that they are climbing a few trophic positions throughout their lives. So they aren't being born into they're probably being born into a relatively high trophic position, but they are still climbing, which is pretty interesting. So how am I going to analyze these results, the ones that you just saw? Well, you just saw 2 bivariate models, the delta 15N and delta 13C values versus the eye lens diameters. Not a very clean version of them, but a version of it nonetheless. We can compare that to other parts of our metadata that we have, looking at total length, looking at the layer types. So if we take out the capsule, if we take out pre-apoptotic, how does that affect the data and stuff? Is there much difference in isotopic values between those different types of layers? We can also start calculating trophic level or trophic position to then incorporate into our other metadata. So that is what I'm working on currently, is starting to do some math and build models based on the isotopic information that we have to calculate their trophic positions. And there's a lot of different thoughts on how this should be done, depending on the literature that you're using. And so I'm looking at a few different ones, and we'll probably ultimately build my own that would fit best in my mind based on the literature I'm reading for sleeper sharks, because currently there's no good ones for ones that are at like top of the food web. So that's where we're at there. And with that, there's considerations that I need to think about when we're processing this data. One of those is that layer order isn't necessarily consistent to the layer diameter between specimens. So as I peeled, it's hard to get the same thickness of peels within per individuals and between individuals. So we have to think of simply each layer as a timestamp in that shark's life, and we have to treat each shark as its own individual in a way. So we can use eye lens diameter to sort of make a linear regression of, eye lens diameter to growth rate of the shark, and that can help us create a baseline for comparing that with the rest of this data. In terms of trophic position models, there's other things like trophic baseline corrections and discrimination factors that we need to consider too. certain isotopes and amino acids will stick in higher trophic levels and get flushed through lower trophic levels. So we got to consider that when we're looking at these results as well. All that to say, we're working on calculating models and interpreting it a little bit closer. And you're probably looking at this model and thinking, okay, I see two delta 15Ns that one is glutamic acid and one's phenylalanine. What the heck is that? This is even more bonus content because we did a little bit of compound specific isotope analysis. And so this is going to help us figure out a little bit more about what's going on within delta 15N. So compound specific isotopes are looking at the isotopes within the amino acids within the organism. So it's getting a little bit deeper. And when we did processes on this, we ran the whole gamut. So we had a lot of amino acids that we can look at. For the sake of delta 15N, we're looking specifically at glutamic acid, which is considered a trophic amino acid. And so what does that mean? A trophic amino acid is going to compile itself more over time through trophic positions. And so you expect it to climb more. And then you can compare that with phenylalanine, which is what we would call a source amino acid, which could also be interpreted as a location sort of amino acid. It doesn't really build up over trophic time or trophic level, but it stays at a relatively constant rate through trophic level is what I'm trying to say. There we go. So on the right side here, you can see a bunch of different groups of organisms within the same region are going to have relatively same phenylalanine level, but their glutamic acid levels are going to be, isotope values are going to be varying. They're going to be significantly different. And we can expect sleeper sharks to fall kind of on this higher end here where we're going to have a pretty big difference between phenylalanine and glutamic acid. And so that raises the question of, is the bulk data being driven by trophic level shifts, which would be indicative by glutamic acid, or habitat shifts, which can be notated by the phenylalanine? And it could be potentially both. So as many things are dynamic, it's not usually one or the other. So one little sneak peek here. This is looking at one of our larger individual females, a 410 centimeter individual. And it's interesting because you see more of a shift in phenylalanine. And while glutamic acid does rise a little bit, that's probably just from the shark growing, getting bigger as it ages and, eating bigger prey items. But there is a pretty distinct shift in phenylalanine. It dips down before it climbs back up. So those trophic level shifts are probably coming more from a location standpoint than a full trophic transition standpoint. Hopefully that makes sense. And the compound specific stuff we did was processed at University of California, Santa Cruz at their stable isotope lab. So put their logo down here for that credit. And so, yeah, that is where we're at there. And what are our next steps among doing that with all the other sharks that we have? We want to start incorporating spatial and other biological context into our data, looking at, as I mentioned, those layer type comparisons, as well as individual variation. So how does things like sex, size, and regional difference all interplay with this compound specific and the stable isotope data that we have? And how can we make clear stories with these individual sharks that give us some idea of potentially where they're going and what they're eating? And so progress per our current objectives is we're pretty much done with the first one. We've pretty successfully formulated methods for delaminating these shark eyes. We've gone through many iterations of it. So at this point, we've got a pretty solid scheme down. For the second objective, looking at delta 15N values, I would estimate I'm about 75% of the way through. We've looked at a good portion of it. I still have a bit to do. And for the delta 13C values, I'm probably about halfway. I've been focusing more on the delta 13C, or excuse me, delta 15N than I have the delta 13C values. So once I kind of get closer with the trophic stuff, I'll start looking at more of the carbon source delta 13C values. So that is where I'm at there. I want to acknowledge the rest of the team, especially NPRB people, because none of this would have been possible without them. Will Litch has taught me and Cindy how to do eye delaminations and tips and tricks, and has had some great insight on the biology and, what could we be looking at with these guys. So it's been really cool working with him. Taylor Broek has kind of jumped a bunch of different labs, but has been a part of this project since the um, birth of it, per se, and has helped analyze all of our stable isotopes and sent off our compound specific stuff and has helped coordinate all of that. And again, wouldn't have been possible without him. Beth Meta, she's gotten all of our islands diameters, which is huge. That info is incredibly important for giving context to size and everything else. And that's a lot of work to do. So thank you, Beth. I also want to acknowledge Darcy Neff. I wouldn't be a part of this project if Darcy Neff was not involved. I also want to thank Daniel Stewart, Bruce Buchholz, and Alan Andrews as their other major contributors to the MDRV project and their own relative realms of it. Stable isotopes is not the only thing that these shark eyes are involved in, and so there's been a lot of things going on with this. And last but not least, I really want to thank all of the observers and fishermen who helped collect eyes for this project. We recognize that these sharks, they're large, they're annoying, and they can be destructive to gear. So them taking the time to actually collect eyes for us and all the data is huge. It's incredibly important, and we've been able to get a lot of cool insight out of it. So thank you to them as well. And these are all the affiliated logos for all the people I talked about and labs that we've worked with. Yeah, and that is where we're at. That is a wrap. Thank you.
Awesome. Thank you, Charlotte. Very nice. Okay, questions. I guess, Charlotte, you can monitor the room and I monitor online. How about that?
That sounds great.
Yeah, and I will just ask the committee and Katrin to hold questions until we torture Charlotte later.
Whoopee.
Ton of work.
Go ahead, Chris.
Do you have depth data to go with the catch for each individual?
Yeah. So Chris asked if we have depth data and catch data for each individual. The answer is yes. We have most of that data for a lot of these sharks. There's some that we don't, and usually in that case, we keep them out of the study, and that's part of our data cleaning process. Is we're kind of taking that sort of, if we have that context, great. If we don't, depending on the size of the shark, we'll keep them or say nay. So yeah, we do have it for a lot of them, but not all of them.
Yeah, I was thinking about that because a lot of your catch was up on the slope from the groundfish fishery, the trawl fishery.
Yeah.
Whereas along the coast, the Aleutians to southeast is longline. And I'm not familiar with how deep they fish for longline, but I was wondering if it's deeper than the trawl, if that's associated with the size difference you're seeing in these.
That's a good question. I'd have to look. We do have that depth data between longline and trawl, and that's something that I haven't thought to look at, but we definitely could.
Can you repeat what that was, Charlotte? I don't know about the rest of
Yeah,
the
she was
piece, but
saying that a lot of the fisheries with trawls done on the Bering Slope are a lot shallower than the longline fisheries that are done along the Aleutian Islands and Gulf. And so is that size discrepancy potentially coming from the depth at which the sharks are being fished, for lack of a better term, that they're being caught at?
yep. That's a good one.
Thank you. Go for it, Aubrey.
How long does it take for a layer to be laid down?
That is a great question that we don't know. Yeah, we don't know.
Again, can you repeat?
Her question was if we know how long it takes for the islands layers to form within individuals. Like how long does each layer take to form? And
yeah. Don't we wish, right?
Yeah, that would help solve a lot of questions. So
yeah. And we have a question from Willa.
Hi, how's it going? I was wondering, with each of your layers of the islands, do you like get multiple replicates for your stable isotope work? And then also, do you have like usually both eyes for the shark or just one eye? And like, does that change what you do?
Good question. We have both eyes for most sharks, not all of them. In terms of how many samples we could run, we only did a couple where we had both eyes processed to look at if there's similarities between the eyes or differences between eyes within one individual. Um, can you repeat the first part of your question? Sorry, I totally forgot it.
Oh, just do you get multiple like samples
Oh,
per eye
replicates.
layer
We
replicate?
we didn't really get replicates per eye layer, in part because each eye layer that we were collecting was getting split off for radiocarbon analysis, stable isotope analysis, and some of them compound specific. We were doing at minimum 2 or 3 different things with each eye lens layer, and they were small enough to the point where we didn't really have the option to split them up. Some of them we could have probably done multiple replicates and we still have them, so we could in the future if we get more money and funding, absolutely, which would be a good idea to go do. But yeah, short answer is we don't really have replicates per um layer.
Yeah, that answered my question. I was just, yeah, interesting to hear about how you could get multiple different things from each one.
Yeah.
So,
Okay.
Okay. Pretty
Are we
quiet
good?
room. I think we're good. One
All
more?
righty. Very good. Well, let's thank Charlotte one more time. And I don't know where we are at right now, Charlotte. Are we going to stay here on Zoom? Do we have a different link?
It should be good until 2, I believe, which I don't think it'll time out after 2. So we could just stay on
Oh,
the Zoom link if you want, or I can make a new one.
no, you can. Yeah, that's fine. Just stop the recording then.
Yep. Yes, yes, yes. Where is my mouse? There's my mouse. There we go.
And
All
then
right. Thank
I
you
would
everybody for
say
coming.
everybody but Cindy, Matt,